Hello students. This lab is about treating wood. It's the treatability lab. And here's our apparatus. This is the treating cylinder. This is our gauge tank, as we discussed in the lecture. Here's a vacuum pressure gauge. We have a vacuum pressure compressor pump here that can do either vacuum or there's vacuum on one side and pressure on the other. There's a switch here to switch from vacuum to pressure. This is a trap to prevent liquids from getting into the pump. So what we're going to do is we're going to treat two different species of wood. So I have 4 samples here. This is southern yellow pine, and it is notoriously easy to treat. Here we have Douglas fir, and I think this may be actually heartwood, so it should be very difficult to treat. So we will treat them both, and you have, excuse me, on Canvas you should have your lab worksheet that I'm going to fill out some of these, but you will have your own lab worksheet on Canvas, and with data included. What I'd like for everyone to do is to collaborate and utilize all the data when you write your report so that you can do standard deviations, statistical analysis, okay? All right, so to get started, safety glasses, gloves. Missed this part when you're remote, huh? Okay. Now first, we need to label the samples so we can tell them apart. And we'll be measuring retention and penetration. So we'll be treating with this blue dye. It's non-toxic blue dye. The concentration is 0.5%, so you'll need to enter that on your lab worksheet. And what we're going to do is we're going to weigh the samples before and after, and that will give us our gross absorption. And then we're going to cut them in half. We're going to rip them in half so that we can get a look at the penetration. So because we're going to cut them in half, I need to label them on two sides, two opposite sides. So I'm going to call this one Southern Yellow Pine 1, SYP 1, on both sides. And this one is SYP 2. Likewise, this one is Douglas fir 1, DF1. And DF2. DF2 has a defect. Somebody has stapled it. This is actually pretty common with when they treat wood, they will staple an identifying label on it. So yeah, that'll be interesting to see what happens to penetration in those spots. Okay, now we need to measure the samples because retention is the weight of the active ingredient, in this case the 0.5%, divided by the volume, right? So we need the volume. So let's get SYP1, and let's measure the length. And I get 15.2 centimeters. And we can measure the width and the thickness. (1917 millimeters), 1919, 1910. 17, 1919, 1910. And thickness. (18.71). 1862. 1918. And the initial weight for SYP1, 33.27. All right, SYP2. 15.2. 1859. So we'll measure it 3 times, take the average. So you'll have some estimate of the error, 18.7. 18.82. And the other direction. 18.94. No, 18.52. No, it's this way, right? 18.93. 19.10. 1901. And the weight, 33.57. Douglas fir one. If it sounds like I'm shouting, it's because the microphone set up here is not best. 15.2. That's centimeters. 1875. 1879. 1883. That's millimeters. And this way, 1759. 1748. 1739. And the initial weight, 22.2. Lower density, huh? Douglas fir number 2. 15.2. Width 1858. $18.60. $18.67. Other direction. $17.62. $17.43. $17.35. And the initial weight, 22.15. All right. So we have the data before treatment, and now we're ready to treat. So we will take samples, load them into the cylinder. To get into the cylinder, you just open this cam lock and slide them in. There we go. Make sure that's locked. All right, move this out of the way, just in case. Okay, so now we want to do a almost full cell treatment. So the full cell treatment, you recall, is we pull an initial vacuum, we then fill the cylinder with the treating fluid while we hold the vacuum, and we fill the cylinder while we hold the vacuum. And we're going to fill the cylinder by using this tubing into our storage tank. We open this valve while we have on a vacuum. It's going to fill up here, it's going to fill up the cylinder, and it's going to fill up here in our gauge tank. Although we're going to use the weights before and after instead of using the volume on a gauge tank, as they do in a commercial treating facility. So we'll bring it up here, close the valve, and we'll open. To get the vacuum, first of all, we put this on V, and we open this valve here, and then the vacuum will see, you can see that, maybe you can't, hopefully you can see that this pump has two leads to it. There's two tubes that go into the switching valve here. So we have one tube that comes out. So both the vacuum and the pressure are going to enter the cylinder here. So when we open this valve, we'll be pulling a vacuum. Right? Here's a gauge that measures both vacuum and pressure. So that's how we will track what's going on. So we'll fill the cylinder, and we're going to wait 5 minutes. We're going to pull a vacuum for 5 minutes. Then we're going to fill the cylinder while we hold the vacuum. Then we will break the vacuum. We have another valve here. You just push it, and it will release either the pressure or the vacuum coming through this valve or pipe. So we hold the vacuum for 5 minutes, and then we release it. As we fill the cylinder's full, gauge tank's got fluid in it. We release the vacuum, and we apply pressure. We'll apply pressure for 5 minutes, and then we will stop everything, drain the cylinder, and remove the wood and see what we've got. Okay, so let's get started. To start the pump, we simply plug it in. And we should be on vacuum here. This valve is closed. So if we open this valve, we should get a vacuum in the cylinder. And we see that we do. And I will take a picture of it so you have a close-up. That looks like 19 inches of mercury. So you can convert that to whatever you like in terms of bar or torr or Pascal or whatever. Oh, and I forgot to start the timer. So it's going to be approximately 5 minutes. You've got the video, so you can figure out the exact time, assuming that the video is real time. And now we just wait for 5 minutes. I'll see you then. Okay, it's been approximately 5 minutes. Now comes the exciting part. We're going to fill the cylinder with our treating fluid by opening the bottom valve, inserting this into the liquid. Opening the bottom valve, the vacuum will suck the liquid up into the cylinder into here. We want to be careful we don't suck it all the way up into the pump or into the valve or whatever. So, be careful. So here we go. Nice and easy. You should be able to see blue here. You should be able to see blue liquid come up through the tubing. Yep. There it comes. Notice that the wood floats. This is true commercially too. That's why they tie it down in the cylinder by some mechanism. And let's stop. All right. I'm going to drain what's in the tube here so we don't get into trouble. There we go. Now, if you look close, you'll see that there's an air bubble in the top. So I'm going to carefully lift the whole apparatus and try and get the air bubble up into the gauge tank. There we go. That's more or less got it. There's a tiny one right there. And now we want to release the vacuum So I'm going to close the valve to the vacuum pump. I'm going to open this valve. You should be able to see the vacuum release here if you look closely. That should do it. Now we switch to pressure. I'm getting my watch ready. Shoot that up again. Let's switch to pressure. Now we're going to have air pressure. The air pressure is coming up to this valve. And this is down. So when we open this valve, we should get air pressure coming in here. And you should be able to see the liquid in the gauge tank drop. And we'll look for that. And again, like I said, we're going to press for 5 minutes. Okay? So this is closed, so it can't get out. And away we go. Looks like it dropped a little bit. We're still coming up to pressure. So you can see the liquid is dropping. It must be going into the wood. And we are like we're stabilizing around 64 psi. So 64 psi, I will take a picture so that you can have a close-up and see for yourself. '66. And I forgot to start the timer again, so we're going to have approximately 5 minutes. So all we can do now is wait. So let's wait, and I will be back in approximately 5 minutes. Let's see how far down this is. All right, it's been approximately 5 minutes, and our gauge tank, I don't think it's moved in a while. So it's time to stop. So to stop, we want to close the valve. So we're isolating the system. Now turn off the pump. Go. Release the pressure. All right. So now we drain the tank, put everything back in our storage tank. Not everything. To do that, we need to open this valve. I need to switch hands here. So we open this valve, and we open this valve to let some air in so everything can drain. I'm holding this lower than the cylinder, lower than the bottom valve, so things can drain. Get this all the way open. And it looks like we have drained everything. Put this in here. Lift this up a little bit. Try to get some of the liquid drained. All right, let's close this. There. All right, time to find out what really happened. So we will open cam lock. Got one drop dripping out. And now we remove the wood. This can be difficult because the wood swells as it fills up with water during the treatment. It swells, of course. So sometimes it swells and sticks. Done this time also. There we go. The issue is the cam lock here is narrower than this tube, so it gets caught. I'll work on it. See if we can get it. There we go. So let's get some absorbency here. And it can be difficult to see the. It can be difficult to see the. Label. Well, that's one. Yeah, the label's there. It's just the label is there, but the sample is so blue, it's hard to find the black label. This is Southern Yellow Pine. I think it'll, we let it sit for a while, I think we can figure it out. Now the ones in the back are a little tricky, so I have my handy tool. For every job, there is a perfect tool. There we go. Let me get some fresh paper towels here. That was for one, I do believe. And sample. Southern Yellow Pine 1. And so on this sample, if you look close, you can see, I hope, that there is some liquid. So I dried it off, right? But there's some liquid oozing out of the sample. So that's called bleeding. We mentioned that in class in a previous lecture. So what happens there is we had it under 60 psi, and then we released the pressure, But it takes a while for that pressure to equalize. And as it does, it can push the liquid that's inside out to the surface so it bleeds. So you can see a little more coming up now. So we'll just give it a little chance to do its thing, and then we'll weigh it. So I believe this is Douglas fir 1, and I think this one is Douglas fir 2. I got that. So here's Douglas fir 1. Douglas fir 2. So let's weigh it to get a final weight so we can calculate the gross absorption. 35.62. Douglas fir 2. 35.62. So it went from 22 approximate grams to 36 approximately grams. Douglas fir 1. 31.27. And it started out at 22 approximately grams. And both of our southern yellow pine are still bleeding. And you might notice here, I'll try and take a picture of this with my camera, it appears to be some untreated spots here on the surface. I suspect those are pitch pockets because the pitch is very hydrophobic and it's not going to treat, water's not going to penetrate. So that could be the explanation for that. And this one's still bleeding. This one's easier to read. Southern yellow pine 1. And it's a little bit of bleeding, but not too bad. 59.15. And we started at 33.27, so that's quite an increase in weight, more than the Douglas fir for sure. And this would be southern yellow pine 2. It's hard to see. Got them really blue. Yeah, southern yellow pine 2. I mean, process of elimination, right? That's what it has to be. 59.50. All right, so collected our data for retention. So you have the dimensions to calculate the volume, and we will, we want the dry volume, right? So we want the volume before treatment, because if we were doing this commercially, we would dry these before we sold them. So the proper volume to use is the dry volume. And you have the weights before and after, so you can calculate the gross absorption. You know that you have 0.5% active ingredient, so that you can calculate the retention. So the retention should be covered. And what we'll do now is I will go rip these on the bandsaw and come back. And I'm not going to take the video camera with me, so you can watch me cut these on the bandsaw. So I will be back as soon as I get the job done. See you in a bit. All right, I'm back. Here are our samples. Get some. Southern yellow pine number 1. This is the outside. This is the inside. And Southern yellow pine number 2. Again, complete and total penetration. This is Douglas fir number 1. Outside, inside. So now we have to figure out the penetration on the Douglas fir. And this is Douglas fir number 2, outside and inside. So notice that we did get significant penetration here, but it's all longitudinal. So you recall the longitudinal permeability is much higher than tangential or radial. And you see that we did get a little bit of penetration across the grain here. So, and also here are our staple marks. I don't know how deep they went in, but it's very possible that this stripe treatment here is due to the incising that we basically did with the staple. The liquid was able to go in and then move longitudinally. So the value of incising is also shown. All right, so what I will do now is the southern yellow pine I'm calling good. 100% penetration as far as I can tell. Except for I don't even see the pitch pocket anymore. So stuff has diffused in there. There are areas that are lighter and areas that are darker, but all the areas that I can see are penetrated, and I'll be taking a close-up photo of this and including it in the lab folder. So now we must deal with the Douglas fir. This is Douglas fir number 1. I will just use the ruler. There's no way to do this accurately. Now in the, you recall, when they do this commercially, they would have a larger piece of lumber, like a 2x4 at least, and they would bore into it. They would extract the boring and then test that for penetration. So at least for that boring, they can be accurate, right? Because it's only 1/4 of an inch. Here, we've got a different issue, so I'm going to call this penetration. It looks like it's going, I have to just guess what the average is here, and you'll have the pictures, so have the pictures and I'll put the ruler next to it so that you can maybe deal with it remotely. It looks to me like this one is about 2.5, and this one is about the same, about approximately 2.5 longitudinal and less than 1 millimeter tangential and radial or cross section. Okay, so penetration on Douglas fir number 1 is 2.5 longitudinal and less than 1 millimeter, so 2.5 centimeters longitudinal, less than 1 millimeter tangential and radial. So our other strangely treated zebra treatments, in size, whatever it is, it's going to be a little more tricky. So again, I'll take both sides are pretty much the same. So I'll take, this one's not as much. It's about 2 centimeters on that end. And maybe, I'm calling it, yeah, it's about 2 centimeters on either end. But you do have some tangential and radial penetration here, along with that's about 2 millimeters. It varies from less than 1 millimeter to 2 millimeters now to maybe actually 5. Less than 1 millimeter to 5 millimeters penetration, tangential and radial. And very similar on the other side. This is a deeper penetration. This here we got about 4 centimeters on this end and about 1.2 centimeters on the other end. So we had Douglas fir number 2 over here. Penetration was about 2.5. Let me do that again. Douglas fir number 1. Yeah, it was 2, that's right. Douglas fir number 1 is approximately 2 longitudinal and less than 1 millimeter tangential and radial, right? And Douglas fir number 1 Douglas fir number 2, excuse me, was where we had 1.5 to 2. 1.5 and 2 centimeters longitudinal, and we had less than 1 to 5 millimeters tangential and radial, plus a stripe, right? Which could be due to incising. Or it could be all longitudinal. We don't really know. I like the incising hypothesis, but these are not enough data to substantiate it one way or the other. And okay, so now you have retention, you have penetration. You've seen the process that we go through to treat the wood. So you should be able to write a good report. Use the template that is on Canvas and submit your report on Canvas. And that concludes this laboratory. I still need to clean up, but I'm not going to record that on video. And I will see you in class. Thank you for your attention.